Hello, everyone. I'm Meredith Ashby, the Director of Microbial Genomics and also Cancer and Immunology Strategy here at PacBio. And I want to welcome everyone to the PacBio webinar series with today's topic focused on using PacBio sequencing to understand SARS-CoV-2 and the host immune response to infection. It's my pleasure to introduce today's speakers who will be lending their expertise to the discussion. Melissa Smith, joining us from Mount Sinai Icahn School of Medicine, and Corey Watson from the University of Louisville, Kentucky. We have a lot of great material to cover today, and the presentation portion of the webinar will be followed by a Q&A session. You're welcome to submit your questions at any point during the webinar by typing them in the question tab on your dashboard. We'll be recording the webinar and making it available for download in the next few days. I also wanted to point out that we provided you with 3 useful handouts that you can download now also from your attendee dashboard. So with that, I will start my presentation on the internal work we've been doing here at PacBio on sequencing the SARS-CoV-2 genome. And I'm going to pause my video so that we can all focus on the slides. Okay. So a number of protocols and primer sets have been published to help with sequencing the SARS-CoV-2 genome. And I wanted to point out that we have posted a wide range of these, including both the primers and the PCR protocols on our website at pacb.com/COVID-19. And with those downloads, we have both the originally published primers and also modified M13 tag titers to facilitate barcoding. So today I'm going to talk in a little more detail about work we've done verifying the Mount Sinai sequencing protocol and also a protocol from the Eden lab in Australia using slightly longer amplicons. So I mentioned the M13 tagging, and so for those of you who aren't familiar with our barcoding methods, this involves adding an M13 short sequence to the target-specific sequence in the forward and reverse. And so then when you've done the initial first round of PCR, you amplify, of course, the targeted sequence. And then those M13 tags allow you, with a second PCR step, to then add a sample-specific barcode using those M13 tags to amplify the target sequencing. And with this method, you can barcode and multiplex up to 1,024 samples on the PacBio system. And there's a detailed protocol for this also on our COVID-19 webpage. So for the Sinai protocol, this approach uses 2 sets of tiled amplicons, a 1.5 kb set and 2.5 kb set, and used together, those provide good coverage of the genome. So this is executed in a total of 4 different multiplex pools, and you can see how the amplicons are tiled across the genome here. So this is our results from doing these amplifications in-house. The top 2 panels here, what you see is that we broke out those pools and did all of the PCR steps individually to check that we were getting amplification of all of them. And then including with the M13 revised primers. And then we went back to the multiplex pool. And here you see the first round of PCR, again, using the M13 tagged versions. And then the second round of PCR, we added a barcode. And so you'll see you get nice, good results from both of those, from all of the pools. And I just wanted to reiterate that these are all done with published PCR conditions. And for this work, we have been using, we started our development using the twist synthetic control. And then when the ATCC heat inactivated Washington strain became available, we switched to that. And so you can see here, all of the regions were successfully amplified with 25 PCR cycles. So here we have a view of the coverage. So this is downsampled to 1,000 reads per amplicon because we sequenced just the one sample on the Sequel II system. So there's a lot of data. And so you can see again the amplicons at the top and then the coverage below. And you'll notice that for most of these, we are getting full length amplification of those primers. And one thing I want to point out here is that you can see 3 SNPs very clearly. And these are expected because what we've done is we aligned the Washington ATCC control against the Wuhan Q1 reference. So these are 3 known SNPs that we are able to call very easily. And then I will just note that we are continuing to work with this protocol in-house to do some primer rebalancing to reduce coverage variance among the amplicons. So the second protocol we have been working with is the Eden 2.5 kb protocol. So here you can see the schematic from the Eden publication, where there's 2 sets here that are tiled. And once again, we first did each of the PCRs individually, so there's a total of 14. And we did this again with the M13 tag versions of these primers. And then in the protocol, these were presented as singleplex reaction, but we went ahead and we attempted to pool these in the way that was alluded to in the paper. And as you can see, you get nice amplification, either with the original primers as published, and then also with our M13 tag versions. So you will notice that with the pool A, you do get some side products with the M13 tag version, but these can be cleaned up with 2 ampere purifications to get a clean result. And so here you can see the bioanalyzer trace after the ampere purification. And then on the right, what I'm showing is the overall coverage distribution across the Wuhan reference. So you'll see that there is some variance, but actually the variance is pretty good. It's only about a factor of 10. And so we think that we can improve upon that by doing some primer concentration rebalancing. So we did a little bit of analysis here. So once again, we downsampled to about 1,000 reads per amplicon, and once again, mapping the Washington control data to the Wuhan reference, and then analyzed it using our minor variant tool, Juliet. So once again, we are recovering the 3 ground truth SNPs. And so a nice thing here is that you can actually download the subsample data set from the link here. And if you want to hear more detail about the analysis, you can register for Smart Leiden, where our bioinformatician Liz Tsang is going to be going into a lot more detail about variant calling on SARS data. And you can find Smart Leiden at pacb.com/events. There's one additional strategy that we are looking into, and that's target-based capture using IDT probes. So this work so far has been done, again, using the Twist synthetic control. So since it's a synthetic control, the first step for us was to add poly-A tails, and then we're basically following the iso-seq protocol where we make cDNA and then do probe capture of the cDNA followed by sequencing. So here you can see the coverage. You'll note since the Twist synthetic control is in several pieces, you do see this 3' bias effect for each one of those pieces. And so that's just an artifact of the control that we're using. And once again, Liz is going to go into a lot more detail with this on her Smart Leiden talk, so I encourage you to register for that and hear what she has to say. So the next steps for us, as I mentioned, we'll be doing some primer rebalancing with these protocols. And then second, we have reached out to some collaborators, and they are going to be testing both of these against patient samples that they have with a range of viral titers. And the idea there is to ascertain the coverage with real-world samples, and then also to assess the success rate of the different length amplicons with non-cultured samples, which may be somewhat degraded. And then finally, I want to point you again to our COVID-19 website. The protocols are here. We'll be posting revised versions of these, including the multiplex pooling of the Eden protocol later this afternoon, and we will continue to post more data there as it becomes available. And then last, I want to acknowledge all of the people inside PacBio who've been working hard to present this data, both our scientists and then also PacBio security and IT, who have been helping everyone with remote work and also is getting into the building. And I'd also like to acknowledge Twist for gifting us the synthetic control so that we can get started on this promptly. Okay, so without further ado, I would like to introduce Melissa Smith, who's going to be talking about HLA sequencing. So from the very start of her scientific career, Melissa has been focused on immunology and virology. She did her PhD in virology at Harvard Medical School, studying how broadly neutralizing antibodies arise during HIV infection. She then traveled to France to study immunology at the Institut Pasteur before returning to her roots at the Center for AIDS Research in San Diego, developing NGS methods for sequencing HIV-end. Her enthusiasm for DNA sequencing led her to briefly join us here at PacBio, where she developed and promoted methods for using PacBio to sequence full-length viral genes and also the immune response. She is now an assistant professor at the Icahn School of Medicine, where in addition to running the genomics core facility, she studies the genomics of complex variants and populations with a particular focus on virology and immunology research. Over to you, Melissa.
Thanks so much, Meredith. I really appreciate the introduction, and to all the organizers at PacBio for allowing me to present some of our work today. So as Meredith mentioned, I'm the Associate Director of Technology Development here at Mount Sinai. Our aim is to use novel sequencing data to address complex problems in genomics and transcriptomics. To do that, we use advanced instrumentation in combination with hybrid experimental approaches and a lot of novel molecular methods to target the detection of low-frequency variants, characterize heterogeneous samples, and elucidate complex genomic structure. We do that through a large suite of different technologies, although today, of course, I'll be focusing on the work we've done on the PacBio instruments. So for those of you who haven't thought about HLA since immunology class, I want to give a quick refresher. So HLA is the human leukocyte antigen, which is just the human name for the MHC complex in our genome. The HLA proteins themselves interact with the T-cell receptor in order to activate and modulate adaptive immunity. There are three main classes of HLA, although today I will focus on class 1 and class 2. So class 1 is expressed by most nucleated cells. Here you can see it represented coming off of this yellow target cell and represented by these yellow protein building blocks. It binds an antigenic protein here and presents it to a T-cell receptor in order to stimulate activity. It specifically interacts with CD8 positive T cells, and when presented with the appropriate secondary signaling, triggers cytotoxic T cell activity to clear anything that is thought by the body to be non-self. Class 2, on the other hand, is presented by professional antigen-presenting cells, and by those I mean macrophages, dendritic cells, B cells. And in this case, the class 2 complex presents their antigenic peptide to T cell receptors on CD4 positive T cells, and when again stimulation goes according to plan, stimulates these types of CD4 T cell helper responses, including cytokine production, B cell help, etc. So because the body needs to be prepared to encounter and respond to any antigen possible in the system, there's a huge diversity of HLA alleles that are mixed and matched in every one of us. And that constitutes the HLA loci as being one of the most polymorphic in the human genome. And certain alleles or allotypes are represented, overrepresented or underrepresented in certain populations. This figure was taken from Janeway's Immunobiology 4th edition. So at the time of that publication, these were the counts of the different alleles of the different classes at that time. For sure, it has increased since then. The reason that's critically important for the conversation we're going to have today is the concept of HLA restriction. So because HLA has this peptide that binds in this binding groove, there needs to be a very nice match between the HLA protein itself and the peptide it binds in order to appropriately engage the T cell receptor. So in this case, you see a peptide that binds with the HLA A0201 allele that engages appropriately with the T cell receptor and leads to recognition and activity downstream. However, if the same peptide was bound to a different allele, in this case B5201, the conformation of this complex is not such that it's appropriately matched to the cognate TCR, and that means there's no recognition and no activation downstream. So specific HLA alleles are capable of binding and presenting very specific antigens, and that cognate matchup is important for appropriate T cell activity, which is thought of as HLA restriction. And I want to show you just a quick example of why this is critically important. This is a paper published by Mary Carrington in 1999, and it was in the HIV space, but it's very critical for understanding HLA in any context. In the first panel of figures, you see Kaplan-Meier survival curves for patients that either have heterozygous HLA expression at all loci, meaning they have the most broad distribution of HLA alleles expressed and therefore enhance their chances to bind all different kinds of antigenic peptides. They survive longer than those individuals who had homozygous representation in the alleles and therefore restricted their ability to express and bind and activate with a variety of different peptides. And if they looked at specific allele expression in particular, in this case B35 or CW04, you found that these alleles of HLA were actually detrimental, and these patients died much more rapidly than those who didn't carry these alleles. So this idea that specific HLA alleles are important for mounting the appropriate immune response is critically important when you're thinking about how HLA modulation can control response to viral infection. Of course, this is HIV. The topic for today's talk is COVID. So all of us have seen these types of figures about the symptoms of COVID to be on the lookout for. This was taken from the CDC website just a few days ago. So patients, are known to have fever, cough, and shortness of breath. But if you look below this flyer on the website, you see sort of a growing list of alternate symptoms. And anecdotally, in the news and across social media, there's a much longer list of potential linked symptoms. It's also estimated that there's a large majority of asymptomatic cases, although I don't think our testing rates have caught up quite to define that proportion. Such differential responses, though, suggest that there's a critical role for the host in responding to infection. And by response to infection, I mean both adequate, preventative, protective immune responses, as well as the potential for overreactive responses that lead to cytokine storm that can be part of the COVID-19 disease pathogenesis. So we're not the first people to think of this as a potential factor for disease. There are a few papers that are already out there, both published. This one just came out a couple weeks ago in the Journal of Virology, looking at HLA susceptibility to SARS-CoV-2. There's several preprints on medRxiv and bioRxiv looking at the same thing. But the idea of profiling the human immune response to try to understand the relatedness and associatedness of certain alleles and certain human genetic polymorphism with the response to infection is critically important for us to understand what it will take to protect us through a vaccine down the road. So I want to show you a little bit about what we've been doing to do that in our lab. And we're doing that by combining PacBio sequencing with the upstream commercially available and validated HLA protocol offered by the company GenDx. So this is a schematic of their workflow, their NGS-Go workflow on the left-hand side. They have come up with a validated, very robust set of PCR primers for both HLA class 1 and class 2 that come in different mixes and matches that they call AMP-X. Their kits also provide library and indices reagents, but we won't be using those because those are for Illumina sequencing. And of course, today we're talking about PacBio. However, once we do sequence the amplicons that are generated, we do a data analysis with the NGS engine tool, which I'll speak of in just a moment. As I mentioned, GenDx has a repertoire of amplicons to address HLA. 11 loci can be prepared with their primer sets, although we focus on using the 6-plex MX61, which comes as a single tube multiplex. What we do with those amplicons is then put them into SMRTbell Express Prep. So we take the double-stranded amplified material, we ligate SMRTbell adapters, in order to generate a SMRTbell library. This is the sequencing library used on the PacBio system. Once those libraries are generated, a sequencing primer is annealed to the SMRTbell, and then a polymerase is bound and sequencing commences. The advantage of long-read sequencing, of course, is that you go around a smaller amplicon multiple times, which allows you to collapse these reads to generate a highly accurate intramolecular consensus or hi-fi read. And when you map these hi-fi reads to the full-length sequence of the HLA molecules, you get coverage that allows you to phase the two alleles at each position within an individual without imputation or bioinformatic reconstruction. So our protocol is as follows. We take between 200 and 250 nanograms of high-quality genomic DNA, so this cannot unfortunately be done with FFPE material. We do the GenDx M61 protocol as written, which gets you 6 loci, 3 class 1 and 3 class 2 in a single PCR. The amplicons that come out of this are mostly full-length genes, and they range from 3 to 6 kb. These amplicons then are used to generate SMRTbells as previously described, although we do use a barcoded adapter set that allows us to generate a specifically barcoded library per patient. So each of these libraries contains all 6 amplicons that can then be multiplexed into a 48-plex, which we then sequence on the sequel system using the SMRTcell 1M. Whoops. These reads are then generated into hi-fi reads after data is collected using the circular consensus sequencing algorithm. So again, multiple passes are combined into a high molecular accurate intramolecular consensus. And then using the NGS engine software, these FASTA files are used to align, visualize, and HLA type the loci with the mapping of discrete SNPs visualized in this image. So again, we use 200 nanograms total DNA input. We get a very robust amplification from most patient samples, generating around 100 nanograms per microliter of amplified material generated per sample. So that's total from across all 6 loci. This will vary widely per patient and per sample quality. We then use 150 nanograms of the total amplified product as input into that Express Prep V2 sample prep. Here's actually what these libraries look like. So this is a bioanalyzer 12K chip showing the gel image of the amplified pool here on the left and the actual traces of 6 of those samples here on the right. So you can see that there is a site broad distribution, which is to be expected. This is a 3 to 6 kb pool, but there's very little to no noise or smaller amplicons following amplification. It's a very robust, very specific PCR response or PCR reaction. Following SmartBell prep, we equimolar pool each of them into that 48-plex, and we're currently doing that manually, although we're hoping to automate it on the Agilent Bravo quite soon. These 48 plexes then are run on the SQL system using a SmartCell 1M and 20-hour movies. This is actually some data from a recent pool we ran last week. We were able to generate with 148 plex 27 gigabases of total data. This actually was a smart cell that was slightly underloaded. We typically aim to have the P1 value here to be 50 or 60%. So I think in our next run, we'll increase loading concentration and get far more data than that. However, we were quite pleased with this run. It certainly was enough to type these 48 samples, which I'll show you shortly. What's really striking is this curve on the right, which is the base density graph. The polymerase N50 here was over 170 kb, which means that 50% of the data we collected was in reads of 170 kb or longer, which means that if that was an average of a 4 kb amplicon, it went around it many times in order to generate that highly accurate hi-fi read. So we demultiplex the barcodes of this total data set. We generate the hi-fi reads using the default parameters on SmartLink 8.0, so 3 passes per molecule with a base quality of Q20. And then we export the FASTA that is generated by that job and import it directly into the NGS engine software. So this is a snapshot of what that NGS engine software looks like. So when you import your jobs, we analyze using default parameters. The data itself and the software itself maps each of the loci separately and then calls SNPs, coverage, and 8-digit typing, as you can see here. So the mappability here is in this column. So again, we're using well over between 95 and 100% of our reads for this mapping, which means there's, again, very little PCR artifact in the system. We're getting read lengths that are on average between 3 and 6 as expected. The read depth per allele is weighted to some alleles over others, and that is expected. There's going to be some PCR bias, but again, the number of reads that we're getting per allele using this 48-plex, even with an underloaded smart cell, is plenty in order to call these types of very high accuracy and confidence. Then, of course, we get the HLA typing. Because you have that full-length read with no imputation required and the highly accurate data as input, we're able to type these HLA out to 8 digits. It also shows you the mismatch rate, so we have no mismatches from the database in our exons, although we may have some in our introns. And then the phasing regions, which are here, are 1, because we don't need to create phase blocks because we have those long reads that span the entire gene. The other thing that GenDx's NGS engine does really well is produce a very nice PDF-based report for sharing with our clients and collaborators. So this is just an example of said report. It's usually about 10 pages long. You get your typing result if there are ambiguities in the sample and what field they're in. This is what a Class A report looks like. Again, you see the mapping I showed you earlier with the SNPs that are called in the exons and introns. The first graph here shows you the level of base variation. Please note this y-axis is logarithmic, so you actually see the base variation mostly below 1%, which makes sense as our hi-fi reads are Q20 or above. The depth of coverage is at 2000 and is steady across the entire molecule, and our quality value is quite high. Here's an example of the DPB1 class 2 locus, a very similar readout. What I really like too is at the end, it gives you an overall view of the number of reads that were mapped to each locus and how that distributed within your pool and how many of the total reads were used. So we're using 97% of those hi-fi reads, which leads to a great utility of the data for this application. So quickly looking ahead, we are going to continue to process HLA typing from the COVID cohorts we're working with here at Mount Sinai. We have several hundred samples lined up for this type of processing, with the obvious aim of examining allelic distribution and its association with disease severity, viral pathogenesis, et cetera. We hope to expand this multiplex panel to 11 loci. As I mentioned, the primers for all 11 loci are available from GenDx, but you have to do them as separate reactions. So 11 PCR reactions for 11 loci, and they all require quite a large input of genomic DNA. So if there's a multiplex reaction that does come out from GenDx in 2 or 3 tubes, we would jump on that in a heartbeat because we could use less material. We often are very limited by material from these precious samples. We're also hoping to work with both GenDx and PacBio to continue to expand the multiplexing options to continue to drive prices down, perhaps with barcoded primers or increased plexi for sequel 2. There's a lot of options out there. And of course, we'd love to apply similar methods to any research project where research-only HLA typing is necessary. And to that end, we are a PacBio certified service provider. I'm putting my email as well as my director, Robert Sebra, and my sequencing manager, Nancy Francoeur, at the bottom. If any of you out there are interested in pursuing this, we're happy to have further discussions with you. So with that, I'd like to thank our team, many of whom are pictured here, our director, Bobby Sebra, assistant directors, Kristen and Michael Beaumont, as well as Nancy, Betsy, and Irina, who did the work I presented here today. Also, thank you to PacBio and GenDx for being very supportive of this project, and I look forward to taking questions at the end of the call.
Thanks so much, Melissa. That was really great. I feel like I learn more about HLA sequencing every time I hear you talk about it. So now we're going to do a short poll. And so the question is, which applications do you think will be most important as scientists work to understand the COVID-19 pandemic? And you can pick your favorite one. And Melissa scolded me earlier on that I did not put TCR sequencing on the list, and that a lot of you would probably want to pick that. But since that's not a topic for today, I restrained myself to only the things that we're discussing today. When I see everyone is voting. Awesome. So let's see, 41% of you picked viral genome sequencing as the top mark, and I think that's really true, especially we're starting to see now discussions about mutations and how that might pertain to regional differences. And then a lot of you picked HLA, so I count that a vote for the T-cell / HLA immune response. And then many of you also picked either BCR or IGH locus haplotyping, with the remainder focused on other types of variant in the genome. So thank you so much. So now I want to introduce Corey Watson. And maybe Corey can change some of your minds about how important BCR and IGH sequencing is going to be to understanding the immune response. So Corey Watson began his study of immunology at Simon Fraser University, studying with the influential immunogenetics researcher Felix Braden. He continued his studies as a postdoc at Mount Sinai and is now an assistant professor of biochemistry and molecular genetics at the University of Louisville. At the heart of Corey's research has been a drive to make the tremendous variation at the IGH locus accessible to science by improving methods for BCR sequencing and VDJ inference, and also by enabling direct haplotyping of the IGH locus. His ultimate aim is to explore how better resolution of the IGH locus can be leveraged to inform our understanding of disease susceptibility and clinical health outcomes. So I'm going to hand it over to Corey now. I think you're muted, Corey.
I'm muted. I'm unmuted now, hopefully. I wanted to say thanks to Meredith and PacBio for inviting me today to talk about some of the work my lab is doing. And I will also shut off my camera so that you don't have to look at me. And I'm hoping that all of you can see my first slide. Is that true?
Yeah, looks great, Corey. Thanks.
Okay, perfect. Good. All right, I'm going to turn my camera off. So a short disclaimer, I'm not going to be showing you any COVID specific data today, but more talking about the general questions that my lab is interested in as a means to maybe think about the role that some of the methods we've been developing and questions we've been asking might fit into the current pandemic. But generally speaking, we think that the work we're doing has a lot of broad applications and the disease space in general. And so, as you might have already guessed, my interest and the interest of my lab is to study how genetic variation can contribute to differences and antibody responses we observe between individuals. And so with that, I feel like it's always worthwhile to sort of briefly review B cells, although we won't talk really about B cells that much in the talk, that they are where this begins. And so as most of us probably appreciate, B cells are critical components of the immune system, involved in both adaptive and immune processes. And of course, one of the B cells' claims of fame is the fact that they produce antibodies, which are these Y-shaped molecules depicted here. And in fact, antibodies are also themselves critical to the immune system and how the B cell carries out much of its work. And they themselves have a variety of functions in the immune system and have impacts and roles in disease processes. Of course, given their importance to the immune system, they're of great interest in the disease research space. And there's a lot of efforts, both historically and currently, trying to understand variation in the antibody response and how this influences wellness and disease, how we can use this information to think about better therapies and patient diagnoses and prognoses, et cetera. And with the advent of sequencing, the ability to sort of interrogate variation in the expressed antibodies within human populations has really increased in the last few years, and we're learning a lot more. I'm sure even in the context of this pandemic, we are going to learn a lot about how antibodies function. And so this is my only slide on COVID specifically, but this is just to mention that although we have a lot to learn, that there's obviously going to be a clear role for antibodies in this story. The obvious one, thinking about neutralizing antibodies and how they can neutralize the virus and help fight during the infection. But then there's also some evidence out there that antibodies may have a role in the inflammatory response via antibody-dependent enhancement. But of course, there's still a lot of work that needs to be done in that space. But it is clear already in some of the studies that are beginning to come out that the antibody response is certainly variable in the context of SARS-CoV-2 infection and COVID disease between individuals. And I think as we go on, as more and more of these studies are conducted and we do repertoire sequencing, et cetera, we will learn a lot more about the dynamics of the antibody response specifically and how this variation that we observe between people may or may not impact disease severity. And so, of course, my lab is interested in a general sense, what are the mechanisms, what are the factors that contribute to that variability, with the idea that understanding them, we can learn something more about the role of these molecules in disease. And so the formation of antibody, of course, begins, as most of us probably are aware, with VDJ recombination, which I've kind of depicted here in cartoon form, in which a single V segment, D segment, and J segment are recombined on a chromosome within a B cell to be brought into proximity with each other, at which point they can be transcribed and translated into the antibody, which is depicted here on the left, which consists of 2 identical heavy chains and 2 identical light chains. And so the processes here that I bolded, common control diversity, junctional diversity, as well as somatic hypermutation, what happens after antigen stimulation, have been studied a lot. we continue to learn a lot about these processes and their contribution to the antibody response. But I think it's fair to say that an underexplored area in this model is how does genetic variation, or what we often refer to as haplotype diversity, within the antibody gene regions themselves contribute to the story. And this is in fact what my lab is hyper focused on, and what the majority of our work is based around. And so I'm going to talk to you a lot about this today. And the idea that we have basically is, if we can understand some of these genetic determinants to the repertoire, that we can better define or maybe even predict what a repertoire might look like in a given person, and understand better how these signatures associate with functional readouts, and whether or not that can be used to inform clinical care or understand disease risk in a clearer way. And so there is good reason to suspect that genetics does have a role. I could go through a lot of them. I'll give you what I think is a very convincing one now, and we can talk about others as we go on. But this is a study that came out by Jake Glanville and colleagues in 2011, in which they looked at antibody repertoires in monozygotic twin pairs. And so this is some data showing the gene usage or expression of different V segments. And as you can see from this plot, you can see variation between the twin pairs, which are shown by the blue and red bars, as well as variation between genes. So I just put this in here just to kind of give you an impression that when we look at something, even this sort of basic signature of a repertoire, there's variability between people, and the repertoire themselves has variation within it. But the sort of most significant finding from the study was that when the authors looked at repertoires in monozygotic twins, they found that their repertoires were much more highly correlated than if you look at two repertoires from unrelated people. And so this suggested that there is a very strong heritable component to setting up this baseline repertoire. And the questions we have, of course, is what are those genetic factors? How do we define them? How do we characterize them? And then can they be used to inform our understanding of disease processes, or what the antibody response is going to do in the disease context? So that question that I just posed there has actually been really difficult to get at. And part of the reason is the locus in which, or the loci in which these genes reside, is quite complex. So I'm going to spend a few slides kind of going through these loci and try to impress upon you the complexity that we observe both at the genomic and population levels, and sort of demonstrate why this has been a challenging problem. And so this is the IGH heavy chain locus, which I've drawn here in cartoon form. And so depicted are a bunch of different boxes here, each representing gene segments. So the V gene segments are shown in green. The blue, the segments are D segments, and then there's a few J segments down here at the bottom in yellow. And then the light blue color are the constant region genes, which are the sort of effector functions for the molecule. So the V, D, and J segments encode the antigen binding parts of the antibody. So these are the parts of the molecule that people often focus on when they think about a response. And so as you can see, these genes are all repetitive, and they reside in this one location in the genome, and they are phylogenetically related, and we'll come back to that as we go on. And just for context, the light chain genes are encoded at two different loci in the human genome, the lambda and capta locus, but you can see for context here that they do look similar to the heavy chain. And so it's been appreciated for several decades now that like we see at other immune loci in the genome, there's a great deal of allelic diversity within the coding segment. So this is a plot showing known alleles that are curated within the IMGT database for all the different V segments. And you can see that there's variability across these V segments, with some genes having a lot more alleles than others. What has only become recently appreciated is that we actually believe we have a pretty poor understanding of the extent of diversity that's out there in the human population. And we now know that much of what we do know is biased toward Caucasian populations. And in recent years, as more sequencing is being done, we're discovering more and more alleles with every individual that gets sequenced and characterized. And so it will be interesting to see in the coming years exactly how much more discovery there is to do. In addition to allelic variation, as I already mentioned, the locus itself is complex. So I showed you this cartoon version before of just functional genes, and these functional genes are not the only genes in the locus. They're actually accompanied by pseudogenes as well. And so in the entirety of the VD and J region, there are about 130 to 150 some odd genes within a given haplotype. And what this basically means is that the locus is incredibly repetitive. And in fact, greater than 50% of the sequence within this 1 megabase region falls within what we would characterize as a segmental duplication or repeat sequence, which means that for many high throughput approaches, it's quite difficult to interrogate. In addition, we know now at the population level that there are structural variants or copy number variants. In fact, there are many that have already been characterized at the sequence level in the population. And these can be quite large and contain multiple genes. They range anywhere in size from 9 to 60 kb. And this means that any given haplotype compared to another can vary by tens of genes potentially. And so this, of course, is also a challenge when thinking about doing genomics, particularly when we think about how we utilize existing reference assemblies. In the case of the 2 reference assemblies that we have currently, both of these miss genes or don't include genes that are known to occur in the human population. And so to sort of drill down on some of this complexity, this is one particular region in which every sample we've characterized a haplotype, we see structural variances and differences in gene copy number. And if we break this down, what we actually see is that all of these copy number events are mediated by these 4 complex segmental duplications that sit in tandem. Each of these 25 kb segdupes are highly related to each other with respect to sequence identity, at times greater than 90 to 95% identical. So this again is obviously a challenging region to interrogate. So at the population level, we do know that the frequencies that some of these structural variants that have been interrogated so far are in fact variable. So this is just some data from several years ago looking at populations across the globe for 3 different structural variants. And we see at all 3 of these that in fact there is population level variation. And so when you take the structural variation and allelic variation and sort of put them together, what we create is this complex picture in which if we look across individuals, and this is data from expressed AirSeq data where people have inferred haplotypes. If we look across individuals, we see that for the most part, everyone is carrying different haplotypes. And in this case, particularly in the figure on the right, none of these haplotypes are the same among these individuals. And so we now have a very good appreciation that, in fact, IGH is probably incredibly variable, but the extent to which this is true, we really don't have a good handle on. And a lot of that has been limited by the genome technologies available. And so this is sort of stepping back and looking at the locus as a whole. We also see that IGH maybe kind of stands out when we compare it to the rest of the genome as a whole. And so this is data from 3 completed haplotypes that we've characterized fairly deeply now, looking at the levels of SNP density in the locus relative to other regions of chromosome 14. And what we can see is that on average, IGH is about 3-fold higher in these haplotypes than what you observe elsewhere in the genome. And of course, this variation is not uniform across the locus, and some of the regions are a bit more variant dense than others. And so what does all this mean? And so the big issue here is that despite the fact that we understand that these regions are really complex and that there is variation in the population, we have a sort of poor set of tools to go in and interrogate these regions. And this has actually had and continues to have impacts in a variety of ways, thinking about how we conduct disease association studies reliably, how we use this information or don't use this information to develop diagnostic and therapeutic strategies, as well as even doing basic biology, trying to understand how these genes are in fact regulated at the genomic level. And so a lot of what my lab is thinking about now, or has been thinking about for the last many years, is how do we improve genomic resources in this region to better facilitate a lot of these different initiatives. And so I want to kind of go through a couple of examples of why the lack of genomic resources is a really detrimental thing. And so the first of this is thinking about this idea that we have a very complex locus, and oftentimes we think about representing complex loci with a single haplotype, and in IGH this is inherently problematic. So in this case, we're taking a hypothetical sample which on one chromosome has an insertion and on the other has a deletion. And in this case, we would be comparing this to a genome assembly that has a deletion in it. And so what do we do with the reads from the sample? We, of course, try to take them back against this reference assembly. And in the instance in which there are locations in the reference assembly that match the sample, this is, well, maybe not a non-trivial exercise, but it's a doable exercise. However, in insertion sequence, we often have a conundrum because these reads will end up getting either mapped incorrectly in the wrong position, or we throw them away entirely and we don't collect any information from that region. And in fact, in IGH, using standard approaches, this is exactly what happens because of all the structural variation. In addition, we know the complexity has also impacted how GWAS has been done using microarray technologies, often for much of the same reason, that placing targets for microarrays can also be challenging in repetitive sequence. If you look at the performance of two commonly used microarrays that have been at least used a lot in GWAS studies, we can see that these actually do a fairly bad job of tagging variation within V segments. So this is looking at known V segments across the locus. We can see at R² about 0.8 that in all of the populations that we looked at in this particular study, there were less than 50% of those variants were actually tagged effectively. And of course, the implications of this, you could imagine, especially for immune system disorders, would be great given the number of GWASs that have been conducted over the years. And this is sort of an egregious example of how that carries through. So this is looking at the immunoarray, which is a specific microarray meant to target genes in the immune system. And specifically, this array is sort of, it's fed from GWAS. So GWAS hits will feed into this array. I don't know that it's used that much anymore, but just to give you an example of how bad things can be. So I'm comparing here the coverage of HLA and KIR to IGH. And what we see is quite crazy, I think. Basically, in HLA, we see something around 9,000 SNPs covering the locus. In IGH, we see 5. And so this is obviously not going to do much good at all when you consider the fact that even between two haplotypes, we can see, thousands of SNPs between them. And so 5 is obviously not going to represent the locus very well. And so those are just a couple of examples of the reasons that I would argue, and that my lab argues, there's a need to better genomic resources in this region. And so the question is really, how do we do that? How do we overcome this complexity to basically end up in a world where we can do IGH haplotyping better? And so I think you could probably guess what some of the things I'm going to tell you are, but one of the big arms of my lab right now is actually just creating more reference assemblies. So both of the reference assemblies that we currently have are Caucasian in origin for the most part. And so one of the things my lab is doing, although I'm not going to show data from today, is using PacBio sequencing. We're trying to generate additional reference assembly resources for these regions from multiple different ethnic backgrounds so that these can serve as a better foundation for genomic analyses. And then what I am going to talk to you about now is a tool that we've been developing to enable more high throughput genotyping of the IGH locus and how we're using that to sort of begin doing some of the biology that's been intractable up until now. And so we have basically been developing for the last few years a custom panel, a Nimble Gen Roche panel But instead of using the standard Illumina-based approach, we have been trying to push the libraries to larger sizes in the 5 to 8 KB range so that we can better leverage long-read sequencing with PacBio. And of course, the advantage there being that with the complexity of the locus, long reads offer improved performance for mapping and assembly than obviously short reads do. And so this is work that we've been doing with Melissa, who you just saw speak about HLA, and Ali Bashir, who used to be at Mount Sinai, is now at Google. And then 2 really talented graduate students, Oscar Rodriguez and William Gibson, who have been developing the bench and bioinformatics protocols. And so the first sort of step of this process has been thinking about, I've been harping on this idea that one reference assembly is not enough. And so one of the first things we did was try to figure out how do we incorporate some of this known haplotype variation into a better reference assembly. And so we sort of created this thing that we call the Franken reference, which is basically taking known structural variants in the population that have been characterized at nucleotide resolution and integrating them into the assembly so that we have a much larger target space and then space for mapping and assembly analysis and interpretation. And so this is shown here where we've basically taken a reference and stitched in the sequences representing the structural variants. So we use this to guide our design of the targets and as well as use it to analyze the data downstream. And so the sort of analysis side of things, our goal here has been to create a pipeline that can generate haplotype-specific assemblies, leveraging both PacBio subreads and hi-fi reads, although we're moving more toward just using the higher quality hi-fi reads. And the idea here is we can create assemblies for both haplotypes, which allow us to much better resolve structural variants and then call SNVs and do gene and allele annotations off of those. And Oscar has been working really diligently over the last few years now to package this up into a formal pipeline, which he's called iGenotyper. And so I'm not going to go through all these figures, but it is now, we're getting closer to having an automated pipeline to process these data and kind of have these nice output reports with all the variant information, et cetera. And so in doing this, we've had to do a lot of benchmarking, and a lot of our initial benchmarking started using a haploid cell line, mainly because we had previously characterized this cell line using a back assembly approach, actually many years ago now, and Sanger sequencing. This sample is actually what now stands as the reference assembly in GRCH38 for all of the IG loci. And because it's from a haploid cell line, it represents a nice gold standard reference. So we use DNA from the sample to do our initial benchmarking. And this is the figures that are shown here. I'll try to summarize them quickly. I'm happy to answer more detailed questions after I finish. And so these figures basically show that we get ample coverage of the region, including over the gene segments. When we put this through iGenotyper to generate assemblies, these assemblies represent greater than 98% of the bases in the ground truth assembly. And the base call concordance between our iGenotyper assembly and the gold standard, or excuse me, and the ground truth is greater than 99%, including 100% recall of all of the genes and alleles. We have also gone in to do benchmarking in diploid samples. And so these are 2 samples selected from the 1,000 Genomes Project. Again, we chose these because we had a lot of orthogonal data that we generated in-house, as well as what's available for the 1,000 genomes. And what we show, which is now available in a preprint I should have said earlier, is that especially when we use and leverage parental data, we can basically create scaffolded assemblies that are haplotype specific across the whole locus. And this allows us to, obviously identify novel structural variants, as well as identify VDJ events, which have been suspected to occur in 1,000 genome samples for a while now. And we show because of haplotype assembly, we can basically identify these and sort of remove them from causing problems in the analysis. And again, when we take this data and compare it to orthogonal data, in particular, large insert phosmid clone assemblies, we see that we have high base pair concordance between the 2 assembly approaches, greater than 99% again. And then this is just sort of bean counting, showing that when we translate these into variant calls, that we see high concordance between the phosmids and the parental data. And again, so this is a demonstration that, and I think you can see that if you build upon this, what the power of it can be. This is looking at these two samples again, thinking specifically about just the V allele segments. You can actually see that that, as we talked about in earlier slides, the haplotype variation is quite extensive. So again, none of the 4 haplotypes shown here are the same. So they differ by both structural variants and allelic variants. And of course, in doing this, as we expected, we're identifying novel alleles. So just in these 2 samples alone, we identified 16 novel alleles that aren't currently known in the IMGT database. And so we expect that number to continue to increase, as I said earlier, as we sequence more and more samples. So we also did, we obviously wanted to compare our performance to see how we lined up with other approaches. And so we took our CHM1, the haploid sample, and asked if we took an Illumina data set from the same sample, how does it perform relative to iGenotyper and PacBio capture? And what we see when we compare this to a ground truth SNP set is that iGenotyper basically is highly concordant with the ground truth SNP set with very few false positives, in contrast to Illumina, which has a high false positive rate and a lower true positive rate than we observe for iGenotyper. Likewise, we've been interested, especially because of the GWAS implications that I talked about earlier. We wanted to look to see how iGenotyper, how all the vetted genotypes in iGenotyper align with the 1kg project. And in fact, what we see when we look at variant call sets from the 1000 Genomes Project in one of these samples, 19240, that it actually misses a lot of the vetted genotypes in iGenotyper, a significant amount, and also overcalls a lot of false positives. Again, the implications of that being that if that's a panel, if those samples are used as imputation panels, that there could be downstream consequences, and I'll come back to that in just a second. And what we see basically is when you look in the regions that only iGenotyper is calling, these are actually in regions of the genome that 1000 Genomes had project to determine to be inaccessible. And so what I think we're showing here is that just by leveraging the longer PacBio reads, we're able to sort of gain access to parts of the locus that previously were not being interrogated very well or accurately. And so again, coming back to this GWAS, we wanted to also look specifically at a real life example. So we took a sample, this is in collaboration with Tom Parks in London. We took a sample from their recently published GWAS, I guess it's a few years ago now, in rheumatic heart disease. And we took their directly genotyped SNPs, microarray SNPs, and their imputed call sets. And we asked how did iGenotyper calls align with those. And in fact, it's quite striking. So we used several different hard call thresholds for the imputation just to sort of get an idea of the spectrum here. And basically what we see is that iGenotyper calls in a lot more SNPs in general than the microarray and the imputed call set, as you might expect. But even when the imputed call set calls a relatively similar number, we see that those don't align very well with the iGenotyper SVs. And in general, the SNPs are biased toward the end of the locus. And this is what you see for a lot of microarrays. For whatever reason, they tend to be biased towards the distal part of the locus. And so finally, one of the big issues, obviously, in thinking about this is their scalability. And so we wanted to test different multiplex conditions to understand how this might scale. And so we took one sample, 12878, that we sequenced, and I showed you data for a few minutes ago, again from the 1000 Genomes Project. And we asked what would happen if we basically multiplex this sample and split it up into a bunch of different runs, excuse me, a bunch of different samples. And so all the way out to a 40-plex. And the take-home message from this slide, I won't belabor it too much, is that as you would expect, as we go up in plex, we do decrease coverage across the locus for the assay. And the number of SNVs that get detected do decrease. But the key is that the false positive rate remains really low. So although as you go up in plex, you screen fewer SNPs, the SNPs that you do screen remain accurate. And so I think as we move into being able to use and leverage a sequel II, we'll be able to realize much better and more comprehensive call sets that could be used to sort of augment current GWAS or disease association studies. Okay, and so in the final minutes, I'm going to sprint to the finish line here, as I know I'm approaching time. And so I wanted to give you a little indication of some of the things that we're working on now in my lab, thinking about, okay, now we've developed this tool, how can we use it to better understand the antibody repertoire and the variation we see in the response? And so we wanted to begin by basically asking some simple questions about, can we find correlates in the genome and to the repertoire, much like you would do in a standard EQTL study. And this is motivated by work that we did several years ago, looking at a specific polymorphism in IGH that is known to be linked to influenza anti-stem neutralizing antibodies, in which we looked at the gene IGHV 169, which is known to vary at both allelic level as well as in copy number. And so individuals have many different alleles in the population, and individuals can vary between 2 and 4 copies in their genome. And what we showed with this, although I'm not going to show you all the data from that study, just the one that's relevant to what I'm going to talk about next, is that when you looked at those variants and you compared the way that that gene 169 is expressed in the repertoire, that for both the copy number variant and the allelic coding variant, we saw a strong association with usage, suggesting that these were basically like any QTL that you'd see elsewhere in the genome. So that's one variant in the locus, or rather two if you include the C and V and S and V. But we know that there's thousands of SNPs across IGH. And so what we're doing now, although I'm just going to show you some pilot data today, is really trying to expand this out and say, okay, can we define the functional variants that contribute to variation in the repertoire by leveraging our PacBio assay? And so the sort of basic model that I'm showing you today is fairly simple. Just can we link variants to differences in V gene expression or usage? And so we have a call set now from 50 people. It's our pilot data set. In these 50 people, there's almost 6,000 IGH SNPs among them after some filtering. And then we have repertoire sequencing data for these same individuals, basically collecting information around 57 B segments. And so a quick high-level summary is that if you use a sort of nominal cutoff of -log10 p-value of 0.4, we see that over half of the V segments have at least one SNP that's associated with their usage. And in many cases, we see genes that have lots of SNPs associated with them. So I'm going to show you, just for the sake of time, I'm going to show you 3 sort of examples from this data set just to give you an impression of what the data look like under the hood. And so this first one is the gene 461. And so I'm showing you the association results for this gene in a Manhattan plot. So all the positions are along IGH on chromosome 14 are shown, and each point obviously represents a SNP that we assayed in these samples. And all the places where you see gaps, those are SV regions, which we haven't looked at in this particular cohort yet. And so what you see here is that as you approach this region of the chromosome towards the distal end, you see this sort of peak popping up for 461. And the most associated SNP I'm showing you, box plot 4, showing you that it does indeed associate with usage. And so that's sort of a kind of a simple or basic case that we've observed, and there are many others like that one. And then there's ones that are a bit more complex. And so this example, we noticed the association for the gene 3-9, which is actually quite high. So I should point out, this is only 50 people. The -log10 p-values are really high for some of these associations already, just in such a small cohort. And so what we saw with this one was 3-9, but we also noticed 3 other genes were also associated with the same SNP. And in fact, when I went back and looked at the data, what we actually see is that this SNP is tagging a structural variant, which involves all 4 of these genes. And so this is why you sort of see this inverse correlations taking place. And then coming back to 169 again, we also see this interesting signal at the distal end where you have lots of SNPs kind of associating with 169. this SNP that I'm showing you is not the one we have identified previously. This is actually a non-coding SNP. This region is definitely complex because it has structural variation in it as well, and we're basically seeing that there's an interplay between structural variation, coding variation, and non-coding variation. I'm not going to go through all the details of that, but just to point that out there, that this is actually a very complex region. But what we observe here is that not only was this SNP associated with 169, but also inversely correlated with 2 other genes in the locus, as well as a couple others that are over 200 kb away. Again, suggesting that likely this region could be some sort of master regulatory region in the locus. And so with that, I'm going to abruptly end and say that I hope that you maybe have a newfound appreciation for how complex the IG loci are. I wanted to impress upon you that although there isn't a lot known about IGH variation or IG variation generally in disease, this isn't necessarily because there isn't anything to be found, but in fact, we haven't been doing a good job of looking. And given the prominence or the role of antibodies in the immune system, it would be surprising that there aren't more connections to be found. We just need to do a better job. And so ways that we're remedying this are obviously, as I've said, creating better genomic resources to facilitate such work to take place. And in doing this, we're already beginning to uncover connections that are functional, and we expect that to continue. I showed you some data from a pilot study looking at repertoire connections. We're expanding this out and including disease samples, including COVID work. And I think and I hope that the work we're doing will ultimately help us better understand variation in antibody response with the potential to help us improve clinical outcomes. And so with that, I'll end, and I'll thank lots of people who have been involved, all pictured here, in particular, William Gibson and Oscar Rodriguez, the 2 PhD students who have been leading the charge in developing the capture assays and doing the assemblies, as well as Melissa and Ali, who I've worked with for many years on developing this. It's been a long road. We're finally getting somewhere, I feel like. And then Wayne Marasco at Dana-Farber, who has worked really close with us on the clinical side and infectious disease side, and Tom Parks, who is our kind of GWAS partner in crime. And with that, I'll thank my funding sources as well and be quiet. And thanks to you guys for your attention.
Thanks, Corey. That was really terrific. So I wanted to do a little bit of Q&A. If Melissa and Corey, if you wanted to turn your video back on, that'd be nice. Certainly you don't have to. Sure. So I'm going to start plucking questions from the audience. So the first one is from Ellen Paxinos. Hi, Ellen. She asks, how well do the different versions of the SARS-CoV-2 amplicon sets work on patient samples? Will you do primer balancing and reaction optimization on real samples or using the synthetic control? Yeah, so that is a great question, because obviously, you know, if we do the primer balancing on a control, then that has more to do with how the primers are interacting in a reaction. On the other hand, it could be that in patient samples, parts of the virus are more likely to break down than other parts, and so we might want to optimize for that as well. So I guess I'm going to say that we're going to do both. We can start right now optimizing the primers using the cultured virus control that we have, and then when we get results back from our collaborators on patient samples, what we'll do is we'll do a comparison of the coverage for that, and then also the coverage for the control sample, and see if there are differences. Okay, let's see here. A question for Corey. Are you able to analyze the haplotypes at the single cell level, or does that even matter?
Short answer is no, mainly because of the material that would be needed to get that would probably be difficult. We need a decent bit of DNA to go into the assay. So in principle, you can analyze haplotype variation on the single cell level using repertoire sequencing data. So I'm not sure what the utility of that would be exactly, but it could be done with, I think, yeah.
So do you see B-cell repertoire sequencing as being complementary then to IGH haplotyping?
Oh, for sure, yeah. I mean, I think, so we already use them in combination, as I was alluding to in the final bit of the talk there. But they can complement each other because you can do some inference from repertoire data that you can also use to help inform the assembly process and vice versa. So part of the repertoire analysis is aligning things to reference gene sets. And so with the iGenotyper PacBio approach, you can actually create an individualized gene set that you could then use for repertoire analysis, which would you know, basically enable better somatic hypermutation inference and the like. So they are definitely very complementary, for sure.
Are there plans to create IGH reference genomes outside of the US?
Yeah, so, well, right now we're the assembly project we have at the moment is looking at, we'll create assemblies for multiple African samples, two Asian samples, and 2 more European samples. But then as we expand to the population level with applying the iGenotyper approach, yeah, we'll be looking in all populations across the world that we can get our hands on, basically.
Can I jump in really quick? I actually was muted. One of the strengths of the approach that Corey's developed, Corey and I have developed, is that we can look at the non-coding parts of these loci as well. So some of the imputation methods, Corey method, where they take the B-cell repertoire, impute back the germline sequences, don't see those areas, and it SNPs in those regulatory sequences that can affect B-cell receptor and antibody expression. And so being able to link the DNA and the RNA data is going to give us a lot more insight into the regulatory networks that inform and modulate the antibody response.
Awesome. Let's see, can the iGenotyper work with long-read RNA-seq data like iso-seq?
No. Well, I mean, yeah, no is my short answer.
Okay, so it's not really for expressed, rearranged PCR. It's really just for the germline.
Yeah. Yeah.
Okay. But Corey, can you use it with long-read genomes?
Yeah, in principle you could. We haven't really done any testing on that, but in principle you could, yeah. And I think it would be really useful for, you know, some of the new hi-fi data sets that are coming out.
Very cool. Let's see. Is there any substantial advances in the IGHV study with the epidemiological distribution of COVID-19 across the globe? And is there any hints of racial variation worth taking note of at this time?
I mean, there's my answer is going to be hand wavy. So we know that there's lots of variation between human populations. So I talked about the gene 169, or mentioned it several times. And so that particular gene's really interesting. And we've done some work in the context of influenza, as I mentioned. So the one variant actually that provides those particular broadly neutralizing antibodies with their function, and in the case of influenza, actually varies considerably between human populations. And so the sort of, I guess if we want to call them good and bad alleles, the good allele is at really high frequencies in African populations, but at much lower frequencies in South Asian populations. And so we see individuals in the South Asian population up to almost 50% that don't carry that allele at all. And that allele meaning that if you don't have that allele, you're not going to make broadly neutralizing antibodies using that particular gene. So we're in the process of doing a lot of follow-up work on that study at the moment, but I think there's no reason to expect that there's not going to be other examples like that as we move forward.
Awesome. Do our panelists have any thoughts on what regions of the genome might be responsible for the Kawasaki-like syndrome in infected children? Yeah, so that's a really interesting question, and was thinking about that myself. So Kawasaki-like syndrome has to do with the inflammation of the blood vessels, which is appearing in a small subset of children who recover from SARS-CoV-2 infection. There are some indications that Kawasaki syndrome has some alleles that are, that predispose one to that. And the genes that are known to be involved with Kawasaki-like syndrome involve downregulation of the T cell immune response. So, this is the kind of study where I think it would be really interesting to do a whole genome structural variation detection to look for, and SNP detection to look for linkages outside of some of the obvious, candidates like, the immune response, BCR, IGH, HLA. I would be super interested to see if the same variants pop up or if there's other parts of the genome that are relevant. Melissa, you're nodding your head. Maybe you want to chime in too.
It sounds like the right approach to me. I don't have any further insight. I mean, it certainly is fascinating, and as a parent of a small child, terrifying. So it would be nice to know what's going on.
So I could add something to that. It is notable that one of the probably 10 or so GWAS hits in IGH includes Kawasaki disease. So
there you go.
It was actually one of the first GWAS hits in IGH. So one of the first disease associations in IGH in the sort of modern genomics era. So I think IGH is probably going to be somewhat important in that story.
Okay, so then since we are over time, I want to be respectful of everybody's time and move on to just some closing remarks. First, I want to thank Melissa and Corey for your time today and sharing your expertise and your perspectives on immunology. And then I wanted to briefly mention a few things. We have recorded the webinar and it'll be available for download in the next several days. So keep an eye out for a follow-up email that we'll send to you with a link to the recording. Second, you can stay up to date on other PacBio webinars by visiting and bookmarking pacb.com/events, and that includes the Smart Leiden online conference where Liz will be talking more about the data analysis of virus sequencing. And then finally, immediately following this webinar, you're going to get a very brief questionnaire. We would appreciate it if you would take 2 minutes to fill it out, as it helps us develop content for future webinars. And again, thank you so much for joining us today, and we hope to see you again at a future event. Take care and have a great day.
Thank you.
Thank you.